the last part of the lecture, number 4 of Maryam Kavousi, and that is about research design, confounding, and effect modification.
Welcome back. The final lecture of this series on research design, part 4. Before the break, or in the third part of this lecture, I ask you to think about how to control for selection and information bias in your study, and how to take into account sex and gender informed perspective. If you want to control for selection and information bias in your study, you should in advance think of a very thorough and strict protocol with regard to data collection and subject selection in your study. So this is something that should be designed beforehand, before you start with your study. This should be prevented beforehand and controlled. In selection and information bias, if you have not thought about it beforehand, there is not much room, and actually no room, to correct for it afterwards, after you have designed your study. So you should have a quite clear sex and gender-informed guideline, a protocol for yourself that is purposeful to minimize the chances of bias in your study. Think carefully beforehand about the choices of your study population, the methods of the data collection, all the sex and gender-specific or sex and gender-related factors that can affect participation in your study or that can introduce information bias into your study. Look carefully at sources of case ascertainment, exposure assessment, risk factor information, follow-up assessment, and think whether they can be sex or gender related. If you don't gather information about all relevant factors during the design of your study, you cannot correct for them afterwards. So, think carefully beforehand. Now, I arrive at the final part that I want to cover during this series of lectures on study design and research design, and that is confounding and effect modification. Confounding is the third sort of bias. This is probably the bias that you have heard most about. Confounding happens when a measure of effect of an exposure in your data is distorted because of a third variable. So you have a third variable in your data that's affecting the association of exposure with your outcome, and that distorts the association. So what you're seeing actually is the effect of that variable that you fail to control for. So to be a confounder, a variable should meet certain criteria. It should be associated with your exposure. It should be associated with your outcome. Most importantly, it shouldn't be part of the causal pathway. So it should not be in between your exposure and outcome. It shouldn't be on the pathway and the way your exposure is affecting your outcome. Confounding leads to distorted risk estimate of the association that you have under study. We call it a mixing of effect. So when it comes to confounding, let's consider an example together. This is a study that aimed to study the association of migraine with depression. The investigators found a very strong association between migraine and depression. But what happened when they looked at gender in their study? So when they took into account the impact of what they call gender, actually looking at men and women, let's say, they saw that it happened that women had a larger incidence of depression in the study. Migraine was more prevalent among the women in the study. And actually, when they took into account gender in their study, the association that they observed became less stronger, the association between migraine and depression. This is a very clear case of confounding. How can you deal with confounding in your data? For confounding, there are various ways to deal with confounding in the study design, namely randomization, restriction, and matching, that we will not go over them in this lecture. But also, with confounding, you can deal with it during the data analysis, using stratification, for example, or statistically speaking, you can take it into account in your multivariate analysis. That's also the major difference that confounding has with other sources of bias, namely selection and information bias. Selection and information bias cannot be dealt with during the data analysis phase. You should think about them during the study design in advance. Confounding, you might be able to deal with it in data analysis, but there is a big but here, and that is you should have gathered information about all the variables that you think might be confounders in your data beforehand. If you have those information, you might be able to deal with them during your data analysis. Effect modification is something that you have heard most about when it comes to interpretation of confounding also. Lots of you think with yourself in your analysis, in the study design, in your data that you're dealing with, you think, am I looking at confounding or am I looking at effect modification? So what is effect modification? Effect modification also happens when variation in magnitude of a measure of association, so the association that you're observing in your data between an exposure and outcome, varies based on levels or across the levels of another variable. This third variable, you call it an effect modifier. We call also the effect modifier, effect modification is also called heterogeneity of effect or non-uniformity or effect variation. Also, to test for effect modification, you look for stratified analysis. So you stratify your analysis and you hope to identify the third variable or the effect modifier that is affecting your effect estimates. So now let's look at an example of effect modification together. In this study, the investigators looked at the association of smoking with lung cancer. And they found a strong association between smoking and lung cancer, a risk estimate, a relative risk of 1.7. When they stratified their analysis to men and women, what did they see? So if they stratify, they realized that the effects estimate of 1.7 in total population was 1.1 in men and 10 in women. It is a rather exaggerated example, but what I wanted to show you was that this is a clear example of effect modification, where associations in one group is different from the other group. So the association of smoking and lung cancer here varies across the strata, the two strata being men and women. So you call this variable, this third variable, sex or gender, how you define it in your study, an effect modifier. Now, let's compare confounding and effect modification together. Confounding is the third sort of bias that we already mentioned. And the desirable scenario in your study is to prevent or remove confounding. So you want to eliminate confounding in your dataset. Effect modification, in contrary, is a property of the effect under study that you desire to identify. Effect modification, you wish to estimate it in your data because it can be hypothesis generating. So confounding is the third variable that distorts your associations. What is the action you want to take in case of confounding? You want to remove it. Go back to the association of migraine and depression. If gender is a confounder in this association, you adjust for gender to take away the effect of gender in this association. That's an example of confounding. Effect modification is something that you wish to identify. It is very interesting to see that your overall estimate in the total population is different across the strata of effect modifier, or desirably across the strata of sex or gender. Why? Because it gives you, it can generate hypothesis for you. It can give you some insight also into the biology. It can lead to generation of new hypotheses why these associations are different between men and women. So the effect modification is something that you do not wish to eliminate, but you wish to identify. What do you do in case of effect modification? You do not adjust for it. You stratify and you show the effect across the strata. So it's a desirable, let's say, phenomena or something that can happen in your data. Let's go back to the example of smoking and cancer. If gender is an effect modifier in this association, you want to stratify based on it, and you show the separate effects in men and women. That is something that's of your interest in your data. So you do not wish to eliminate it. You wish to specify it. Here, in the following slides, I've listed for you various scenarios that can help you, that can give you a very quick insight and guide and key to look into your data to see if confounding or effect modification can be present in your data. So what you do is that, statistically speaking, you look at the crude estimate, so the unadjusted estimates of association in your data. And you also look at the stratum-specific estimates. Let's say, for example, the stratum-specific being looking at men and women separately. If there is no difference, so the crude estimate and the stratum-specific estimates are the same, you can say that there is no confounding and no effect modification. If the measures of association are the same across the strata, but they're different from your crude estimate, it can be an indication that confounding is present in your data. And you can deal with it also statistically. If your stratum-specific estimates differ from each other, but when you pool them together, they're similar to your crude estimate, it is also an indication that effect modification is present in your data. But in reality, it's not that simple, because lots of the variables that we know, including sex and gender, can be at the same time both confounder and effect modifier in your data. So when you look at the stratum-specific estimates, they might differ from each other, and then you look into whether both of them are larger or smaller than your crude estimate, and you wish to make conclusions based on that. I've also summarized it here in this slide that can give you a quick guide on how you might be able to look for and deal with confounding or effect modification in your data. So have in mind that it might not be that easy, and they can present at the same time. This is also something you wish to identify. So effect modifier, as I mentioned, is a very interesting phenomenon that can happen in your study. This third variable or effect modifier is changing the effect of your exposure on your outcome. And don't adjust for it. So try to look into your data, try to distinguish the role of confounder and effect modifier, and just don't start on adjusting for everything in your data. But try to think carefully, try to make subcategories, look at the strata if you have enough power to do so. So here I've listed the similarities and differences, let's say, between confounding and effect modification, and why effect modification can be interesting. Effect modification is what we say it belongs to the nature, whereas confounding belongs to your own study. You adjust the confounding to take away this distorted effect from your data set, or the distortion of effect from the association that you're seeing. But in effect modification, it can be very simple and useful to look at the effect modification in your data set. So you want to look at different estimates because it increases your knowledge of biological mechanisms that can be behind the association of your exposure with the outcome. Effect modifier also, besides the biology, can be very interesting in devising public health actions that are tailored to a specific group. So, and these here also I've listed different ways of, let's say, assessing and adjusting for confounding. And also I've listed the ways how you assess and how you deal with effect modification. But please have in mind, you do not adjust for effect modification in your data. Here I've listed an example for you, including 4 different scenarios, A, B, C, and D, where your effect estimates in total population are presented for the association between smoking with coronary heart disease. And I've also presented here the associations in subgroups, stratified by sex. I've made up 4 different scenarios, and I want you to think in this slide. Look at the crude estimate, look at stratum-specific estimates between men and women, and decide for yourself when sex can be a confounder, when it can be an effect modifier, and when it can be both, like scenario D. And I want you to have in mind that it's not always very simple. It's not always either confounder or effect modifier, but you deal with many different real-life examples that a variable, here sex for example, can be both confounder and effect modifier. So how do you decide whether a variable is confounder or effect modifier? I showed you some examples, and I gave you some quick guides on how to look for confounding and effect modification in your data. But do you only purely base your judgment on whether a variable is a confounder or effect modifier on statistical terms? So simply saying that if it is statistically significant, then I call it a confounder or I call it an effect modifier? Or you base it on your subject matter knowledge, on your prior knowledge of the subject under study? I would say combine both. Don't purely base it on your statistical approach. Well, I just told you that if you have a large enough sample size, everything can become significant in that data. If you have big data available, everything can become significant. So big data can be also very dangerous. Not everything, let's say, that you get statistically significant in real life should be regarded as confounder or effect modifier. So always also use your own knowledge on the subject matter under study. Read, and read about what you are studying. So know the disease under study very well. If you want to imply whether sex and gender or whether sex and gender-related characteristics are confounder or effect modifier in your data, you should have a good base of knowledge. So read the previous literature on the topic. It's likely a combination of both. You can combine statistical approaches with your subject matter knowledge, and then decide whether a variable is a confounder or effect modifier in your dataset, or likely both. Now, you've designed your study. You started with your research question. You designed your study, chose the study design. You selected your population. You gathered information. You did your statistical analysis. And now is the time that you should interpret it. You should interpret whether what you found is relevant. So you came up with apparent sex and gender differences in your dataset, or lack of apparent sex and gender differences in your dataset. Both should be interpreted with caution. Remember, have a pre-specified hypothesis. Always think beforehand, what am I searching for? What am I looking for in this dataset? Is it relevant to look for sex and gender differences in this dataset? Have a priori, an understanding of the subject matter under study. Think beforehand about the associations you expect, the magnitude of the associations, and the direction of the associations. Think beforehand whether you expect sex and gender-specific differences or similarities. What is the magnitude that you expect, and what is the direction, based on the previous literature. Interpret the results regarding sex and gender differences in your dataset with caution and in the light of biological plausibility and social context. Remember, cultural differences matter a lot. Also, interpret your results in the light of accuracy. Think carefully and examine your dataset for the possibility of bias and confounding and chance finding, especially when you have a large dataset. So if you have ruled out the possibility of bias, confounding, and chance in your dataset, and you've met all the criteria, then cautiously you can interpret your sex and gender-specific findings in your study. When it comes to reporting your results, the final step, You should be very transparent in reporting your results. You need to report whether you had a priori a pre-specified hypothesis for testing for sex and gender interactions or not. Normally, large databases, the big data, helps you to reduce type 2 error in your dataset. But it's at the cost of introducing type 1 error into your dataset, meaning that you might introduce false positive findings in your dataset just because your sample size is large enough and everything becomes significant. So be aware of the possibility of false positive findings in large datasets. When you're looking for confounding factors, also look specifically for confounding factors that can be associated with and affected by sex and gender. Also, if you find differences, investigate the intermediate pathways or variables that can be, let's say, intermediary in the pathway to understand the apparent sex differences that you're seeing in your study. Be transparent, report it, and specifically report whether your findings are surprise findings, whether you had a pre-specified hypothesis, or whether this was a post hoc explanatory analysis that you did and you found these sex and gender differences. And report, report, report. Make the opportunity for the others to replicate your findings. Even if you don't find anything, put it in your supplementary material. It can be the starting point for the others to start a study based on the results of your study. It's sometimes possible that the journals are not interested in the negative findings, but you can still present them in your supplementary material. Collaborate. If your sample size is not large enough to detect sex and gender-specific findings, increase your sample size by collaboration with other studies, but make sure that your studies are comparable in terms of design and subject selection and information. And last, in this series of lectures, we went over sex and gender considerations in methodology and in your research design. I tried to explain that having a sex and gender-informed perspective is very important in all stages of your research design. I hope to have been able to encourage you to design a sex and gender-informed study that is thoughtful, conservative, comprehensive, reliable, responsible, and the study that lacks bias or minimizes the chances of bias by thoughtful thinking. And remember, lack of thoughtful, thorough study design and methodology can lead to bodies of literature with inconsistent findings regarding sex and gender differences.
So we heard a lot of confounding and effect modification. Lieza, if you look at your research, how do you tackle the confounding in your research? Because if I look at dementia, for instance, maybe there are also confounders.
Yes, I think there's always confounding, but I think we are especially interested in effect modification. And as Maryam already explained, I think stratification is key in research to identify effect modification. So in my current research, for instance, I'm looking into differences in post-stroke cognitive impairment. And we know from literature that about 50% of survivors have post-stroke cognitive impairment. And I compared it between women and men, and it's about the same. But then I looked into more detail in the cognitive domains, and I also stratified that by gender, and I saw differences. I saw effect modification. So different domains are...
so not in sex, but yes in gender.
Well, I think this is complex because it's a sex and gender discussion, but probably it also has to do with educational chances people have had in the past. So we think certain cognitive domains for verbal speech is usually better
Verbal expression.
Verbal expression is better in women compared to men. While, for instance, visual orientation is usually better in males. Also, probably because they've been more practiced with that because of technical occupations or...
but this is really gender specific.
So I think it's gender, but it's I think also still a debate. It could also be sex, biological differences in the brain.
Yeah. So stratification is then key to be aware of effect modification factors.
Yes.
Yeah. Do you have similar experience, Jeanine?
Yeah, well, I've experienced that when I review papers, that very often there's no stratification at all. And that's, I think it's really a pity and a lost opportunity. And sometimes, well, I comment on it and then I get it back in a revised version. And then you do see that there are differences between men and women.
But they didn't notice.
They didn't notice because it's like by default, very many researchers immediately the first step that they make is to adjust for sex and for age. So then they present as their primary result age and sex adjusted models. And well, for example, I reviewed a paper about it was about cytokines. So these are kind of inflammation factors in adipose tissue. And they looked at if they were different between men and women. And then you found a lot of differences. So fat is not similar in men and women. And then it was very interesting to see like which factors were different. But unfortunately, these researchers were already interested in it. So they did not adjust for sex. But if they would have done it, they wouldn't have found anything at all. Because then you adjusted everything, well, flat.
Yeah. Are you aware, if you do your own research, of effect modification?
I know it's possible, but, well, I don't have an example for you to present. But I know that because we just discussed this, that Maryam has very interesting results in genetic data. So maybe, Maryam,
can you give us an example?
So I certainly agree with Janine and Lieza that the default is now adjust for sex and age and sex in your first model. And this is also the practice that was really done in all the genetic studies, what we call genome-wide association studies, up to now, let's say. It's years that we have started huge consortium efforts and collaborations to just pool all the sample sizes together, because for genetics you need huge sample sizes. And everybody just used to adjust for age and sex in their analysis. And just very recently, it's now, GWAS have been there now for 15 years. And it's just last couple of years, people started looking at sex, at both sexes differently. And it's amazing to see how many new genetic discoveries in certain fields, diseases that you can imagine, like cardiovascular disease or anthropometric measures, are coming out now that are specific to one sex. There are difficulties how to deal with it, logistics, like sometimes the imputation of chromosome X so far was not very easy. So, but if all of these difficulties are tackled, I think stratification is certainly a powerful approach to provide us with more biology behind what we are seeing.
More enrichment also.
Yes, yeah. Certainly. But it can explain more of a biology. Having said that, I always advise the students also that if you want to do that, have really a specified hypothesis. And this is what I want to stress. Because if you have a large enough sample size and you start, it's tempting. If you start stratifying, chance finding, I mean, the chance that you find something in one of the strata is always there. And you will overrate your findings just because your data set is huge. And that single, association in that very specific group becomes significant.
But if you do it in a transparent way and report it very properly, other people can replicate
it. Exactly. Collaborate and replicate. I think that's really the main key here. And there's always also a balance. Of course, lots of what we know of a lot of beautiful biological discoveries are really came about by just doing this, like stratification analysis, and sometimes you think it's a chance finding or something, a surprise finding, but that happens to be a strong biology behind it, and others replicate it. Yeah. So there should be a balance between, like what to do and what not to do.
And read. You already said that. But what if I look into your research, Jeanine, you look also at medicine, for instance. In medicine research, to look at male and female in the former studies, it wasn't there. How is that possible? Because it's so necessary to do. And is it possible to look at that data again?
Well, that's a very good question, Maria. And that's the problem, is that recently we wanted to look at differences between efficacy and safety in cardiovascular drugs, and also cardiovascular drugs which are used very often in the population, such as ACE inhibitors. And those studies are already done quite some years ago. And what we wanted to do is we did a meta-analysis, so we pulled all the data together because we knew that the individual power of the studies would not be large enough to do women and men specific stratification. But what we found is that we found more than 30 studies that would be eligible and would be very interesting to look at differences between men and women. But really, none of the studies reported sex and efficacy or safety stratified by sex. And the problem was also that the studies were that old that we tried to...
Weren't you astonished?
I was, because these medications are used by like millions and millions of people. And we don't even know for sure if the efficacy and the safety is similar for men and women. And it's even worse because we know just by pharmacovigilance data is that the most common side effect of ACE inhibitors is a dry cough. And that's reported more by women than by men. So you would assume, and maybe even they didn't know in those first studies, that first trials, that this was the case. But still, if they would have reported and stratification of the data in men and women, and it's not even necessary to do it in the main paper, but even if it was available in the supplemental,
one of the side studies, yes,
that's not a problem. But if it would have been findable because we looked everywhere, then at least we could have said something about it. And even if the studies were not huge, if the data is there, then it's very important to for studies like
to do
what we did,
studies again,
meta-analysis. Yes,
Lieza, do you have similar experience in neurology?
Yes, definitely. I tried to do a systematic review for risk factors, treatable risk factors, and we could identify a lot of papers, but only a slight proportion reported it stratified by sex or gender, and then even vital information was missing about the exposure or about the prevalence by sex, which makes it really difficult because you can, of course, then pool the couple of studies you did find, but then I think there's some bias because other studies did not report.
The comparability.
The comparability, you're never sure why some studies did report it stratified by sex, others did not. What was the reason for this? So I think it's really important, but I think we've stressed it now a couple of times that people really should report it stratified, even in supplementary. It's just a small line, but it really will help to get the bigger picture.
Yeah. Your message to the audience, if it comes to confounding and effect modification, what is your main message?
Stratify.
Stratify. Jeanine, and your message?
Yeah, well, I mean, I would like to add what Maryam also said, is that, of course, stratify, but also think in advance, like, why do you think that stratification is important? Because the difficulty is if you have a large data set, everything will become statistical significance. And then you really have to think ahead, like, why do you think? And I mean, it might be true and it might be not true. That's research, you don't know.
Yeah.
But think of, like, why do you think that this might be an interesting analysis to do so?
Thank you so much. Thank you all three for your knowledge, your experiences on this topic. And of course, Jeanine, good luck with your research, and Lieza also. And Maryam, a big thank to you, because you really gave us a lot of food for thought. So, and thank you all for watching this workshop, Gender in Research. And I hope that it inspired you for your new publications.